Varghese, Dr. Alex George and the research team at our center to determine the cause underlying the cleft lip and palate anomaly, especially genetic associations related to this, and this research has been sponsored by Smile Train.
The cancer research in CMBF focus on the role of sortilin, a sorting protein, and its involvement in the extracellular vesicle biogenesis and transportation. We work on the concept of blood liquid biopsy by studying and profiling the extracellular vesicles in the lung and breast cancers with a future focus on personalized medicine.
In my 8 years of clinical practice in Jubilee Mission Hospital, the JCMR has helped me a lot in the various areas of cytogenetic and molecular studies. I'm also very excited to be part of the new studies in the areas of the liquid biopsy, which is going to be the future in diagnostics of oncology.
I'm extremely delighted to see the progress of our research center. Over the last 10 years, we have crossed significant milestones in Kerala and in the national level in medical research.
The ICMR, Short-Term Studentship, STS program promotes research interest among medical undergraduates, with 27 of our MBBS students securing ICMR grants for their projects.
During my MBBS period here, I had the privilege of working under the guidance of Jubilee Center for Medical Research regarding the COVID-19 patients. We studied the clinical epidemiology and laboratory findings of COVID-19 patients. This paved an important way for effective control of COVID-19 patients in our hospital. And this study was later published in a well-known Elsevier journal. And in 2023, we had the privilege of linking this article to the United Nations Sustainable Developmental Goals.
Providing office assistance and supporting MBBS students, DM candidates, PG students, project interns and trainees.
We have a public health scientist, Dr. Anjali Annie George, who helped the medical faculty to formulate their research, to analyze and lead to publications. We also have a senior scientist, Dr. K.G. Raghu, recently retired from NIST Trivandrum. He is working on various biomolecules.
Cytogenetics and Genomic Laboratory conducts genetics and epigenetics research on reproductive biology, hematological malignancies, breast cancer, and rare genetic disorders.
So identification of genetic variants and its function analysis in disorders of sexual development is one of the major research area in our laboratory. The understanding genetics and epigenetics background of polycystic ovary syndrome is another area of our research. And currently we are focusing inflammatory and steroid pathways in PCOS. So currently 2 external funded research projects headed by Dr. Ravindra Angadil are going on in our laboratory.
This laboratory is equipped with a Zeiss Metasystems imaging platform that is coupled with a high-resolution CCD camera and dedicated software tools for karyotyping and fluorescence in situ hybridization and real-time PCR. In 2021, Professor Dr. Ravindran Angadil, former professor of genetics, University Science Malaysia, had joined our center as a senior scientist. Identification of genetic variants and its functional analysis in disorders of sexual developments is one of our major research areas.
DSD, that is disorders of sexual development, the old terminology being intersex, and in this particular condition of DSD, we normally find there is a discordancy between the genetic, gonadal and anatomical features of a patient. And today we are with a tie-up with the JCMR in order to study the genetic causes for DSD.
Understanding the genetics and epigenetic background of polycystic ovary syndrome is our another area of interest. Currently we are focusing on the inflammatory and steroid genesis pathways in PCOS.
I've been associating with the JCMR with Dr. Suresh in the molecular diagnosis of myeloproliferative neoplasms. around 20 to 25% of myeloproliferative neoplasms have the risk of transformation to acute myeloid leukemia. So we are trying to locate other genetic mutations which can lead to diagnosis of AML, transformation of AML.
In 2019, our team has reported a novel missense mutation gene in a female patient with 46XY karyotype.
One of the recent success stories of Jubilee institutions is JCMR. Here, research is not a different work or added work. It's part of professional life.
We facilitate clinical researchers to prepare papers and help them getting it published. We work in tandem with doctors to submit their research proposals to different funding agencies.
The publication graph of Jubilee has improved steadily with more than 100 publications per year and cumulative impact factor exceeding 150. To promote faculty research, JCMR distributes publication incentives, having already awarded over 15 lakhs rupees.
I have 43 publications to my credit, including 4 chapters in textbooks. One of my papers, which is a registry of atrial fibrillation from across Kerala, have been linked to the United Nations Sustainable Development Goals to fight the various challenges that the world faces today.
Biochemistry Research Lab is focusing on proteomics studies related to chronic inflammatory and autoimmune diseases.
In the process, we are aiming to establish a clinical proteomics domain combining biochemistry and clinical science in pursuit of biomarker discovery in the form of proteins.
Phytochemistry laboratory focuses on the extraction and characterization of secondary metabolites such as alkali and flavonoids from Carica papaya leaf. Our team recently discovered 2 candidate biomarker protein in diagnosing exacerbated chronic obstructive pulmonary disease variant and published the study.
In association with Dr. Matthew John, who's a senior scientist at JCMR, we have discovered 2 new protein molecules which could serve as potential biomarkers for exacerbated COPD patients. And in future, we expect that these molecules would serve as potential biomarkers and would help in clinical diagnosis of different phenotypes of COPD patients.
We are also equipped with immunofluorescence facility to detect autoantibodies in blood and CSF associated with neuroautoimmune diseases.
Being a tertiary referral hospital, we almost daily come across various cases of neuroautoimmune diseases. Clinically, many of these diseases share common clinical features. So just by clinical examination or by other investigations like imaging, it may be difficult to differentiate these entities. For the prompt clinical identification of these syndromes, we require testing of various neuroautoimmune antibodies, which is done from blood as well as from cerebrospinal fluid. With the help of our research team, headed by Dr. Matthew John, we are able to promptly identify these neuronal antibodies, which hugely helps in the clinical diagnosis of the patient, which in fact makes a huge difference, a prompt difference in their final treatment and their outcome.
We have an ongoing ICMR-funded project for the work on lipoprotein multiomics integrative approaches in differential diagnosis of autoimmune diseases. Our study has its broader applications in nutraceutical and pharmaceutical aspects. Recently, 4 novel flavonoids from Carica papaya red lady leaf have been reported from our lab and published the findings.
Jubilee family is really proud of our JCMR because we could go a lot, reach a lot through our research program. We're really proud of our research institute and also those who are working in connection with our research institute.
Jubilee has completed 58 externally funded projects, and there are currently 17 ongoing projects with a budget allotment exceeding 7 crores.
During JCMR, we always explore transformative medical researchers supported by Central Ministry of India. Being part with us not only fuels the vital medical researches happening in Jubilee Center for Research, but also receives 100% tax exemptions under the Section 35(1)(2).
The research culture we inculcated in Jubilee institutions is unique and has no comparison among similar institutions on this part of the country.
The National Symposium on Genetics in Clinical Medicine, organized by JCMR and the Dr. D.M. Vasudevan Endowment Oration, initiated in 2019, to celebrate Dr. Vasudevan's 50 years of academic excellence, have significantly elevated our institution's profile. Featuring global speakers like Dr. M.V. Pillai, Dr. Shyamsundar Kottili, Dr. Gagandeep Kang, Dr. Sumesh Philip Thomas, Dr. K. Valsraj Menon, and Dr. Vinod Scaria, these events foster international collaboration and highlight our commitment to clinical medicine.
The Laboratory for Computational and Structural Biology consists of a group of highly motivated researchers focused on solving complex biological problems through in silico, in vitro, in vivo, and structural biology analysis.
A typical drug discovery cycle will take at least 7 to 10 years of time. If we see a drug came into the market, we have to realize that there are a lot of efforts done by different kind of scientists like medicinal chemists, structural biologists, computational biologists, etc. In our lab, we are trying to merge both the computational biology as well as the structural biology to come up with novel therapeutic molecules for the diseases like Alzheimer's diseases, uterine fibroids, etc.
The lab is working on drug discovery and trying to develop new candidate molecules. We are also unraveling the complexities of the structure, function, interactions, and molecular mechanisms of disease-causing missense mutations. We are designing and evaluating novel progesterone receptor modulators as potential drug molecules for uterine fibroid. Also trying to provide mechanistic explanations for the putative fibroid-causing mutations. This is DHR-funded project.
Every day in my OP, I come across 10 to 15 patients with uterine fibroids. Many of them I have been forced to advise hysterectomy. Reason is very simple. There are medicines available, but they are not so effective, and some medicines are there which create complications. So, with the help of Dr. Dilip from the research, we collaborate together to finding a drug for fibroid in the future. It has been accepted by ICMR and Government of India, and we are very happy our research is going on, and I hope that in the future we will come out with a drug which is effective and with less complications.
At the Laboratory for Computational and Structural Biology, novel therapeutic molecules for Alzheimer's disease are being proposed, focusing on several potent drug targets. This research program is supported by the Spices Board, Government of India. The efficacy of Ayurvedic nootropics on Alzheimer's disease as a part of Chief Minister's Nava Kerala postdoctoral program is also undergoing.
Additionally, the center studies the molecular mechanisms of protein aggregation through in silico, in vitro, and structural biology techniques, advancing drug discovery. Research also includes a multidisciplinary approach to understanding the phenotypic-genotypic relationships of Ayurveda Prakriti.
As part of the decennial celebrations, JCMR organized workshops and training programs in advanced scientific disciplines. These included the CRIST workshop on current methodologies, the proteomics workshop on protein analysis in biomedicine, the cytogenetics fish workshop on genetic diagnostics, and the DNA Watch program on advancements in DNA technologies.
To date, JCMR have successfully organized over 70 journal club meetings, each attended with great enthusiasm by our medical faculty.
Intramural grants are also provided to initiate novel projects until external grants are received.
Research Day celebrates with vibrant participation from both staff and students of Jubilee institutions, which showcase the latest academic research and innovative projects.
JCMR has MOU with different institutes of national and international importance such as NIOH Ahmedabad, RGCB Trivandrum, CMC Vellore, Karunya University Coimbatore, Spices Board Kochi, Riken Japan, Canterbury Christ Church University UK and many more.
Now we have 15 scientists, 23 research scholars, 14 project staff, 5 supporting staff, totally 57 talented individuals to drive our research efforts.
Here at the Jubilee Center for Medical Research, we are more than just researchers. We are pioneers committed to the advancing health of our society. Over the past decade, we have built a foundation of knowledge and innovation. Now, as we look into the future, we are poised to uncover even greater discoveries. We are confident that the years ahead will underscore the vital role of the Jubilee Center of Medical Research in the global medical research community. Together, we will continue to break barriers and illuminate the path of a healthier world. Because at the Jubilee Center for Medical Research, we don't just imagine the future of health, we just create it.
Jubilee Center for Medical Research began his humble journey in 2014 and quickly gained recognition from DSIR in 2015. Our research specialty lies in translating clinical problems into innovative laboratory solutions. Today, JCMR has 4 laboratories and 3 supporting facilities to achieve our cutting-edge research ambitions. The Cell and Molecular Biology facility works on the genetics of cleft lip, dedicated to identifying the root cause of orofacial clefts. We also work on the concept of blood liquid biopsy by studying and profiling extracellular vesicles in lung and breast cancers. The Cytogenetics and Genomics Laboratory conducts genetic and epigenetic research on disorders of sexual development, PCOS, hematological malignancies, breast cancer, and rare genetic disorders. In 2019, our team reported a novel missense SRY mutant gene in a female patient with a 46XY karyotype. The Biochemistry and Phytochemistry division is also equipped with an immunofluorescent facility to detect autoantibodies in blood and CSF associated with neuroautoimmune diseases. The Biochemistry Laboratory has identified 2 candidate biomarker proteins for diagnosing different phenotypes of COPD. The Phytochemistry Laboratory has recently reported 4 novel flavonoids isolated from Carica papaya. The Laboratory of Computation and Structural Biology focuses on solving complex biological problems through in silico, in vitro, in vivo, and structural biological approaches. We are designing and evaluating novel progesterone receptor modulators as potential therapeutic agents for uterine fibroids. We are also working to repurpose traditional medicines for the treatment of Alzheimer's disease. Additionally, we study protein aggregation mechanisms and the phenotypic-genotypic relationship of Ayurveda Prakriti to advance drug discovery. Our animal research facility, recognized in 2015, actively supports both academic and translation research. The Institutional Human Ethics Committee received approval in 2016, ensuring the highest standards of ethical research practices. Beyond laboratory achievements, JCMR has emerged as a vibrant academic platform. In 2017, we successfully organized a national symposium on genetics in clinical medicine, bringing together leading scientists and researchers. Continuing this tradition of academic excellence, we hosted National Conference on Omics in 2024. Every year, we conduct Dr. D.M. Vasudevan Endowment Oration to honor his 50 years of academic excellence. Over the years, JCMR has achieved several significant milestones. In 2017, we were recognized as a research center by Kerala University of Health Sciences and registered with Niti Aayog. We obtained income tax exemption under Section 3512 in 2019 and CSR approval from the Ministry of Corporate Affairs in 2021, highlighting its commitment to social impact. Recently, JCMR was recognized by Manipal Academy of Higher Education for its PhD program. We foster interdisciplinary collaboration to address complex medical challenges and maintain MOUs with institutions of national and international importance. Jubilee has completed 65 external funded projects and currently has 17 ongoing projects with a total budget exceeding 7 crores. As we complete 12 years of remarkable achievements, we invite you to explore the endless possibilities of innovation and collaboration with us. Powered by dreams and driven by mission to transform lives, JCMR is a place where research...
Good morning to one and all, and welcome to Dr. D.M. Vasudevan Endowment Oration 2026. This program was instituted in 2019 in honor of 50 years of academic career of our beloved Dr. D.M. Vasudevan. And this is the 8th discourse. Let's watch a short video to know more about the oration. All the eyes on the screens, please.
Professor Dr. D.M. Vasudevan is the founder director of Jubilee Center for Medical Research of Jubilee Mission Hospital Trust, Thrissur, Kerala. His leadership accomplishment has significantly influenced the day-to-day activities of JCMR. Under his guidance, JCMR has become a recognized center under DSIR, Kerala University of Health Sciences, and Manipal Academy of Higher Education. This center has Institutional Ethical Committee and Institutional Animal Ethics Committee, both approved by Government of India. The research center has MOU with different institutes of national and international importance, such as St. John's Academy, Bangalore, Karunya University, Coimbatore, NIST SCSR Institute at Trivandrum, Center of Excellence in Microbiome, Trivandrum, Vaidaraknam, Thrissur, Canterbury Christ Church University, UK, and University of Santo Thomas, Philippines. The publication output of JCMR has improved steadily with more than 100 publications per year, and the research funding has also consistently increased.
JCMR also obtained income tax exemption under Section 3512 in 2019 and CSR approval from the Ministry of Corporate Affairs in 2021, reflecting its commitment to scientific excellence and social responsibilities. Under his leadership, several major scientific programs have been organized, the latest being AURA, a national conference on omics, a landmark event that further strengthened interdisciplinary research collaborations. Dr. Vasudevan is a distinguished personality in scientific fraternity. After completing his MBBS, he received his MD in biochemistry from AIMS, New Delhi, and FRC PATH from UK. He is an authority in allergy and immunology and has also worked extensively on cancer. Dr. Vasudevan received the prestigious Dr. B.C. Roy Award for Eminent Medical Teacher instituted by Medical Council of India in the year 1992. He was also honored with the Best Doctor Award in Medical Education from Kerala Government and M.M. Ghani Award of Calicut University in the year 1994 and 95 respectively. His textbook of biochemistry for medical students is being followed by Indian and foreign universities, of which Spanish and Russian editions are also available. He has also authored textbooks for dental and paramedical students. He has guided 31 PhD scholars and has more than 285 publications in reputed journals to his credit. After his retirement from government service, he actively served at Kasturba Medical College, both Manipal and Mangalore, and as founder principal at Sikkim Manipal University and Amrita Medical College, Kochi. He continues to share his expertise with Amrita and Agape Diagnostics, Kochi. Dr. Vasudevan was the founder director of Research Institute at RCC Trivandrum as well as Amala Cancer Research Center, Thrissur. His sustained and exceptionally high-impact leadership achievements demonstrated the effectiveness in achieving JCMR's goals and image. An endowment oration was instituted in 2019 to commemorate 50 remarkable years of his academic career. Over the years, the oration has featured eminent speakers from around the world. The first oration was delivered by Dr. M.V. Pillai from US. The second oration featured Dr. Shyam Kotilil from US. The third oration was presented by Dr. Gagan Deep Kang from Vellore. The fourth oration was delivered by Dr. Sumesh Philip Thomas from Canada. The fifth oration featured Dr. Valsraj Menon from UK. The sixth oration was delivered by Dr. Vinod Skaria from Bangalore. The seventh oration, held in 2025, was delivered by Dr. Ashwin Dalal from Hyderabad. Continuing this proud tradition, we warmly welcome you all to the 8th discourse of Dr. D.M. Vasudevan Endowment Oration featuring Dr. Deshratn Asthana from Pyogenix, Texas, USA. All right, let's begin the program. First of all, I request the dignitaries to kindly come forward to the dais and take their seats. I invite Reverend Father Rennie Mundankurian, Dr. D.M. Vasudevan, Dr. Deshratn Asthana, Reverend Father Terran Mullakara, Dr. M.A. Andrews, Dr. Shibu C. Kallavelapil, Dr. P.R. Verghese, Dr. Raghu, and Dr. Matthew John to kindly come forward to this dais. Let's begin this auspicious occasion with a prayer. I request Ms. Sara to come forward. I request everyone to kindly stand for the prayer.
breaks my heart. Lord, I need you. Oh, I need you. Every hour I need you. My one defense, my righteousness. Oh God, how I need you.
Thank you, Sarah. Kindly be seated. Now I invite Dr. P.R. Verghese, Research Coordinator, Jubilee Center for Medical Research, to welcome the gathering.
A very good morning to one and all. Respected dignitaries, guests, faculty, staff, students, and benefactors of Jubilee. It is an honor to me to welcome all of you to this academic festival that we are celebrating for the 8th year. Dr. D.M. Vasudevan is in our heart and soul. With his leadership, we completed 12 years of achievements. All these 58 years of his academic career, Dr. Vasudevan was living in the soul and heart of his students, among his colleagues. And the Jubilee Center for Medical Research is proud to organize the 8th discourse of Dr. D.M. Vasudevan Endowment Oration today. All about him is already shown in the video played just before. First of all, let me welcome our research director, Dr. D.M. Vasudevan, who made this day possible. Welcome, sir, most lovingly and respectfully. I also welcome Mrs. Devi Vasudevan to this function. Welcome, ma'am. The orator of this function is Dr. Deshratn Asthana, CEO, Pyogenix, Houston, USA. Dr. Asthana is a former professor and clinical laboratory director at University of Miami, Miller School of Medicine. He successfully balanced academic teaching, patient care, and research responsibilities within a large medical teaching institution, transforming a financially challenged clinical service into a sustainable and profitable enterprise through strategic leadership and operational excellence. Today, Dr. Asthana will speak on immune activation and exhaustion in HIV-infected patients. Sir, at Jubilee, we have a group of faculty members, different, interested in HIV, patient care, as well as research. One among them is Dr. Joe Thomas. He is a professor of community medicine who has successfully completed an ICMR project in PLHIV, patient living with HIV during COVID pandemic. Now, it is my pleasure to welcome Dr. Deshratn Asthana sir to this program. I request Dr. Joe Thomas to offer bouquet. Hearty welcome, sir. Our director, Father Renny Mundangurian, will formally inaugurate this function. Father is our strength and motivator. Welcome, Father, to this program. Dr. M.A. Andrews, the principal of Jubilee Mission Medical College, is presiding over this program. He is not only an administrator, but a true researcher. Welcome, sir, to this function. I also welcome Father Terin Mullakara, our assistant director, on his absence, because as many of you may be knowing, simultaneously an inspection on cardiac surgery, cardiac transplantation surgery, an inspection is going on. Father Terin is there. I have to welcome our medical superintendent, Dr. Shibusi Kalivalapil. He is also in that inspection program. They will join in short. I welcome all academic persons from neighboring institutions. Also welcome the friends of Dr. and relatives of Dr. Deshratn Asthana, and students, well-wishers, and family members of Dr. D.M. Vasudevan sir. A warm welcome to all. Warm welcome to all our assistant directors. Warm welcome to our vice principals to this program. I am happy to welcome all heads of the Jubilee institutions, faculty, staff and students of Jubilee institutions to this program. A warm welcome to media persons to this program. A special welcome to our JCMR team members who are working hard for the smooth conduct of this program. Special welcome to Dr. Regu, our scientist, a person known to Dr. Deshratn Asthana through Indian Academy of Biomedical Sciences, who will formally introduce the speaker. Welcome, Regu sir. And Matthew John, our scientist, who will formally propose the vote of thanks. Welcome, Matthew. Once again, I welcome all of you, wishing you a great academic feat. Thank you all.
Thank you, sir. Now I invite Dr. M.A. Andrews, Principal, Jubilee Mission Medical College and Research Institute, to deliver the presidential address.
Respected Director of Jubilee Mission, Father Renny Mundangurian, Honorable Orator, Professor Deshratn Asthana, Professor D.M. Vasudevan sir, all other dignitaries in the dais and off the dais. Whatever I wanted to say has been shown in the video and followed by Dr. Varghese in his welcome speech. So I have nothing much to say, but again I formally as principal, I am very proud and privileged to welcome all of you to this 8th D.M. Vasudevan Endowment Oration. As all of you know, Professor Vasudevan has shaped the minds and future of millions of medical students through his research and especially by his textbook on biochemistry, which we are following and has been translated into different languages. At this time, this oration is actually a tribute to his legendary work of Professor Dr. Vasudevan sir. And I request all of you to please stand up to give a standing oration. Thank you. Please sit down. It is our honor to have a great scientist, academician, innovator, enterprise like Dr. Deshratn Asthana as our orator today. He is going to show how clinical research, immunology, clinical medicine, and industry can be integrated. Rather than not only going through research, he has made up a company and he is the CEO of that company is successfully marketing things, and he is a great researcher, and his talk on immune activation in HIV and exhaustion will be definitely giving us a lot of ideas on this emerging topic. And we are very happy that he has accepted our invitation, and we extend our thanks to him as my own and also as that of the institution. I think this inspiring talk will be giving a lot of ideas to the young minds, how to apply industry or balance industry, research and clinical medicine. I hope this oration is going to be a great inspiration for all of you. I will not stand between you and the great orator. Thank you, sir, for accepting our invitation. Let me conclude with this. Before that, I would like to extend my sincere thanks to Dr. Varghese and his team, JCMR, for arranging such a wonderful meeting. Thank you all.
Thank you, sir. To formally inaugurate the program, we will now proceed with the ceremonial lighting of the lamp. I request Reverend Father Renny Mundankurian, Director, Jubilee Institutions, to kindly do the honors. May I request the other dignitaries to join in. Thank you, everyone. I invite Director for the inaugural address.
Hey, very loving good morning. Dear respected Dr. Deshratn Asthana, Dr. Vasudevan, Dr. Andrews, Dr. Regu, Dr. Varghese, Dr. Matthew, the friends and families of Dr. Vasudevan and Dr. Asthana, and distinguished visitors or participants, delegates from different parts to Jubilee, and my dear Jubilee family members. Today, as Dr. Varghese told us, today is an academic feast. Every year it is repeating. So I don't take much time, only 3 points. First is our Jubilee. I have to say one word, since Dr. Asthana is first time in Thrissur and Jubilee. So one word I have to say about Jubilee. This year we are celebrating 75th year of the foundation of Jubilee, Platinum Jubilee, sir. And we are happy that you are a guest to us in this Platinum Jubilee, Dr. Vasudevan Endowment Oration. Thank you for coming. 75th year, a small beginning, and now we have 8 institutions. We have hospital, nursing school, paramedical, research, medical college, nursing college, Ayurveda, and lastly we have started the Jubilee Medical MHA institution. So this is known as a poor man's hospital. Started with a vision to serve the people, to give better treatment as far as with low affordability. Till now we are trying to maintain this line and hope to go on. As you know, we are in a difficult situation. The nurses' strike is going on. Let us see what comes out. Maybe we will be all forced to take next steps, like the expenses like that. So it's Jubilee, and we are happy you are here. Second point is a word about Dr. Vasudevan. There is no need to speak more about the proud face of Jubilee. Wherever I go, when I say the name Dr. Vasudevan, I see the light in the faces of those who know him. Everywhere, wherever I went, when I speak about research, when I spoke about Dr. Vasudevan, they are happy, they respect. Already described his life and words by Dr. Varghese and Dr. Andrews. And really Jubilee is proud that Jubilee is. Thank you, sir, for your presence and inspiring words and guidance. I don't go into the detail we have seen. And Jubilee Research is one of the dynamic research center in India now. We are really appreciated, mainly because of his presence and guidance. I don't go into the detail. I am really happy he is here, and every year we have an endowment oration in his memory. This is the 8th oration. Every year we had eminent personalities. Thirdly, I've heard about Deshratn Asthana. That's the I asked him how to pronounce. Dr. Andrews spoke like Deshratn. Then in lift, he told us he's called Desh, and that word is taken by Dr. Varghese, but he's Deshratn Asthana. Am I right? So gave his father's name, and he's really a gem, a wreck in this field. Especially, he's spreading the light of Indian intelligence or research in America. So he has been in 40 years there, and still a Deshratn of India. I'm happy he's here. And only 45-50 minutes he has, and I hope we'll get a epitome of this immunology for us in this 45-50 minutes. I'm really happy that he's here. With these words and with your permission, I declare 8th Dr. Vasudevan Endowment Oration is inaugurated. Thank you very much.
Thank you, Father. I now invite Dr. K.G. Raghu, Scientist, JCMR, to introduce our esteemed orator for the day.
Good morning, all of you. And today we are here to celebrate the glory of the contribution of D.M. Vasudevan to the medical research of India. I would like to recollect the contribution of the Vasudevan in the biomedical science. I, being a researcher in the field of metabolic disorder and associated complications, I used to visit different medical colleges as well as the Institute of National Importance in the area of biomedical science throughout the world, not only in European countries but also in other countries. And wherever we go, there will be students or the researchers had been working with Vasudevan sir in one of these institutes wherever I go. So when I talk about D.M. Vasudevan, I am here in the Jubilee. When we talk about he is heading the research, medical research in Jubilee, they used to talk about, appreciate the contribution of the Vasudevan, and they talk about at this age he is still active, he is leading the different biomedical research institute for the betterment of the public health. That is the Dr. Vasudevan. His contribution is enormous, spectacular and splendid in the biomedical research. So today you have a special greetings from the fraternity of the biomedical researchers, not only in Kerala but also from all over the world, for your contributions, commitment and your interest in the medical research. And today I can see Dr. Kumari. I don't know whether she is able to identify me. We met at Calcutta for a DST meeting with Dr. Remini was also there. So when I was talking about your presence to the Dr. Varghese, thank you, madam, for your attendance here. Today we have a special guest to glorify the contribution of the DM Vasudevan. That is nothing but a very outstanding a clinician turned businessman who is presently working in US. That is nothing but Deshratn Asthana. So my present duty is to introduce him for the benefit of the audience about his academic credential and research contribution. So when I was asking the chat GPT regarding who is this Deshratn Asthana, even though I know him superficially through Indian Academy of Biomedical Science, chat GPT had given essays explaining his contribution in clinical science, diagnostic, business, public health, operation, etc., etc. So in order to make for the convenience of the audience, we made a PowerPoint, 2-3 slides for the benefit of all of you. So let me start. Today we have a Dr. Asthana. Basically, he is the, Dr. Varghese was talking superficially about his CV. Dr. Asthana is the CEO of the Pyogenix, a company known for its contribution in the diagnostic. And prior taking this responsibility, he was an academician. He has been in a clinical laboratory director of the infected disease. And he is also the professor at the University of Miami, Miller School of Medicine, for about 3 decades. I think I am audible, isn't it? Yeah, but my short height and everything make a problem. And I'll help you. Thank you, Father. Thank you very much. Now I am okay, I think. You can see me very well. Yeah, thank you, Father. So it is my pleasure to introduce his academic credential. Basically, he is alumni of the All Indian Institute of Medical Science, where he did PhD with what outstanding immunologist of that time. And in addition to PhD in biomedical science from AIMS, he also got an MBA. That MBA was helping him to do business in the area of the clinical science. And he has many years of experience in the clinical research, clinical trials, translational research, contract research, and clinical diagnostic. In addition to that, he has participated in the development, standardization, validation of a number of diagnostic assays for patient care. Some of these test and diagnosis systems were approved by FDA. You know, FDA is nothing but the world accreditation agency for the drug development and nutraceutical development for the clinical use. Now Asthana has been managed a symbiotic relationship of his academic, research, and patient care responsibilities in a large healthcare system, which is also a medical teaching institution. He has over 2 decades of management experience in the area of operation, personnel, and leadership, which has been demonstrated by transforming a large clinical service facility now which is running profitable. Once upon a time, this business was lost. Because of his achievement and contribution and his knowledge in operation, he has been made that institute a highly profitable one. In the area of the clinical research contribution, his team has been providing assistance in clinical study design, study-specific diagnostic and testing services, along with consultation on science, patient recruitment, logistics, data management for more than 130 long-term clinical research projects, 600 clinical trials, 75 collaborative research projects, and 27 contract research projects since 1998. All this quantum of the project where he was involved show his contribution as well as his achievement in the area of the clinical science, all of which brought a tangible benefit. Now you see about the importance of the clinical science in India. Now everybody is asking about the translation. So he is an ideal example for a man who is working in the area of the translational research. And he has bought, and now you see, whenever you submit a proposal for the funding, government is asking about the tangible outcome. Development, but what the idea or the skill you are developing should be a tangible, and he is an ideal example for that type of achievement. And research contribution publication, he is the member of the AIDS clinical trial group and a global AIDS program, and his area of research are the pathogenesis, innate and vaccine immunity, immunity reconstitution, antiviral drug resistance in infected diseases, and in particular HIV infection. And he has a 75 article for its credit in a high impact journals. And he has traveled extensively. Morning he was talking about he has traveled all over the world in connection with his business and research. With these words, it is my pleasure to welcome Dr. Asthana for his endowment lecture. Dr. Asthana, please. And he'll be talking on immune activation and exhaustion in HIV-infected patients.
Thank you, sir, for the introduction. I request the other dignitaries to kindly join the audience. It's now time for the much-awaited oration. Ladies and gentlemen, let us welcome Dr. Deshratn Asthana to deliver the oration lecture.
Very good morning to all of you, and thank you for giving me an invitation to be here. I'm thankful to Dr. Varghese, Professor Vasudevan, and our esteemed father, the director of the institution, and the dignitaries and the people in the audience. Thank you very much for your attendance today. Hello? Can you hear me? Are you better off? Or I should use mic. Mic is better? Okay, let me use mic. I'm extremely honored that Professor Vasudevan's oration, I have given a chance to be participating on this one with the rest of the other colleagues before me. And Professor Vasudevan is an exemplary scientist and physician and innovator in respect, and he has contributed to medicine and health sciences that we are honored that he is among us. And also he is a fellow AIMS alumni, just like me. And the one thing which I think we have missed altogether pointing it out, that you might have seen it, he is one of the simplest and humblest person I have met in my life. And especially in this field of science, where we have so much ego and so much that what we have done, what we have achieved, and all that stuff, he stands out to be out of the crowd and shows up that you have to be humble, you have to be simple, you have to be there for the people to make a difference in their education and learning and teaching and all that stuff. So exemplifies with that respect. So I'm really honored that you have given an opportunity for me to come down. And the Honorable Principal is here, and he has shown me some of the work which is being done in the Jubilee Center and the medical research as well as clinical care. And today also happened to be one of the days when I am here as well as they are having inspection. So it's a coincidence, but it's an opportunity that this institution has exemplified, that we have brought so much excellence that the institution is growing and further growing up in there. And you are all part of this growing. And so I think I congratulate you also for being this part of the journey. Without further ado, because I have Dr. Varghese has, and also Dr. Raghu has spoken quite a bit about my achievement, but to be honest, it's very simple. We all start our career somewhere. I ended up in this field, but that was not my intent backward that time. But somehow or other, I think we come across and we have achieved a little bit in the field. So I'm going to talk about a little bit about immune reconstitution and immune activation and exhaustion. And I will use the experiment model, HIV. And I'm also going to, if time permits, I'm going to also use another approach to define this particular phenomenon. We have often not really missed it, but also we are not able to take care of patients in the real sense, because the medicines can do a certain part, but there's certain aspect of the system which are actually we don't address with the medicine, and we need to really investigate. And financially, we are not in a position to investigate each and every patient, but the personal care and knowing that these tools we happen to have, we can really manage our patients better. And I'm going to show you with some of our work in this field that how we have come to know about this thing and how think we can make a difference in the patient's well-being or in betterment altogether in the due course of time. So just to give you a little bit background, the chronic immune activation is negatively associated with immune recovery and positively with progression to the AIDS in HIV patients. And the chronic HIV infection leads to disproportionate levels of immune activation as well as immune exhaustion. And I'm going to explain to you what I mean by immune exhaustion in these patients or in patients in general when immune activation remains at chronic stage. And then persistent immune activation is a consequence of inflammation, and markers of immune activation and inflammation remain abnormally high despite successful therapy during HIV infection. So these days we are using combination antiretroviral therapy, and we may be able to suppress the viral load to virus load in the patients to undetectable level, but it does not really solve the problem because patients are still having replication going on at the base level, and that once kicks in due course of time, it's also going to really impact about their recovery as well as long stability there. Further, association of soluble and cellular markers of inflammation and immune activation and immune exhaustion in HIV infections. I'm going to explain you some of these from our data. And immune exhaustion, which is basically when you have immune activation for a longer period of time, leads to a situation whereby the body has, say, for example, immune systems. And I'm sure all of you, since in the biological sciences, you may be knowing it, that body constitutes of T cells, B cells, and so many other types of cells. Each of these cell types have a function. So when you keep them in a certain stage, in a highly active stage altogether, they eventually lose the purpose. So they get exhausted, just like we get exhausted in our day-to-day life. So the cells get exhausted. When they get exhausted, then even the medicines are helping them out to bring the numbers of cells. We can produce a lot of cells, but actually they are not doing their job. They are exhausted. They are not performing their function, what they intended to be. So I'm going to show you some of the data in that respect. So overexpression of the negative costal molecules, which is basically we have the PD-1 and TIM3 and CTLA-4. These are the markers which are used to define the immune exhaustion in this patient population. And so just to give you a scheme of things, that how the chronic immune activation in HIV patients plays out. And there are a number of things are going on in a given time in a patient. So for example, that we have microbial translocation is also happening at the gut, which leads to immune activation. We also happen to have a persistent HIV infection, which also cause of immune activation. Then in addition to that, the patients also are having to have the co-infections like CMV, hepatitis C virus, and others that also can lead to immune activation. And activation of plasma, cytoid, dendritic cells is another cell compartment which we are getting a interest and significance in as we are learning about the immunity, that also plays an important role in terms of leading to immune activations. And then altered Tregs and TS17 cells, I'm going to talk about a little bit of those ones, they also lead to immune activation. So there are a number of factors, a number of cell types, or a number of systems are working in a given time in a patient, which leads to a stage of immune activation. So even if we are able to control the viral load and say that patient is under control, nothing is happening, but actually there are a lot of things that are happening, which in due course of time is going to really deteriorate situation in the long course. And then what will happen with immune activation and exhaustion, that we have impaired recovery of CD4 T cells, which are affected in HIV infections. And that could be basically the cause where patient from HIV infections lead to the AIDS, which is the clinical condition of the patients in that stage. So what do we know so far? And I'm going to share, before I go to the in-depth of the science, that CD4 T cell subsets are constituted of Th1 type cells, Th2 type cells, Th17, and regulatory T cells. These are all part of the CD4 T cells, their number of cells. So we are going to talk about how we looked at these cells and how we identified which cells play an important role in this process. Then IL-17 is producing Th17 cells involves in inflammation and the tissue damage. And that is also a good marker to understand the impact of pathogenesis in these patients. And particularly in autoimmune diseases, it has been well studied. Recent studies have show that role of Th17 cells in inflammatory response in chronic viral infections in a number of different diseases. I think I moved it. Move first. Can you go back? Okay. Yeah.
So backward this one.
Backward.
3, 4 times.
So it is previously reported that IL-17 is produced mainly by activated CD4 T cells. Th17 cells are known to be involved in pathogenesis of different inflammatory conditions. Their role in HIV infection is not well investigated. And Th17 cells in circulation have been linked with inflammatory response in various infectious and non-infectious disorders. So we analyzed IL-17 producing CD4 T cells in a group of HIV infected patients which have been on the combination antiretroviral therapy along with the pro-inflammatory Th17 cell phenotypes in the memory CD4 T cell compartment. And we determined the association of disease progression markers like immune activation and immune exhaustion with these cells. This is the study participant. All my work has been on the actual patients, not the experimental models which we used. So what we are describing is actually real patients which we have recruited in the study and identified and studied them over the course of time. And they have been, as I was mentioning back there, the viral load is below detection level. So So less than 50 copies of virus was at the time undetectable, considered to be undetectable. So these patients have been clinically under control. So they are not, basically we can say that they are having the active infections at the time, but the same token they are HIV infected. And so here, this particular slide, I'm trying to demonstrate that the relationship of immune activation, exhaustion, and microbial translocation in HIV patients. And as I talked about TS17 cells, so there are two types of TS17 cells. Peripheral TS17 cells, which are germinal cells, and then we have CD4 specific TS17 cells, which are activated CD4 cells. And here we are looking at this particular graph, that the relationship of activation markers, which are defined by HLA-DR and CD38. These are like in general identified markers of immune activation in the patients. So we compare back when we happen to have the direct correlation between TS17 cells and the activation markers. So the higher the TS17 cells, the higher the activation markers, as you can see in the graph 1A. And then the graph V talks about the relationship of peripheral TS17 cells with the soluble CD14, which is the markers of immune exhaustion. And this is also a positive relationship, or direct relationship altogether between these two. And then we look back the relationship of peripheral TS17 cells with PD1 and TIM3, which are the markers of the microbial translocation. Sorry, the exhaustion. Here we look back CD4 positive IL-17 cells. I told you that is IL-17 cells are two types of cells. So this will look back the same parameters, and that whether they, like this particular compartment of cells, are having that same relationship, that higher the immune activation because of the TS17 cells are growing up with the inflammation there with patients, and that what proves to be that's the case, as you can see from this particular graph. We look back also the soluble CD14, which is a marker for microbial translocation on the IL-17 cells, and that's the same, have a direct relationship there. And then we also look back the relationship of these particular cells with the markers for exhaustion, which is PD1 and TIM3, and that also have positive relationship there. So the point I'm trying to make it that the higher the inflammation, the higher the activation, and that is also happening with the defining by the exhaustion as well as translocation. So all of these factors are playing an active role in a given time in a patient. In this particular slide, we look back now CD4 T cells, and we look back in the context of Tregs, which are T regulatory cells in HIV patients. And these cells play an important role in kind of regulation of this compartment of cells, which we talked about, that CD4 cells are constituted of TH1, TH2, and TS17, and the Tregs. So Tregs play an important role in terms of looking back the various aspects of triggering the action in different cell types. So here we are looking back if the Tregs we look back in the context of, say, TIM3, which is marker of immune exhaustion, the higher the exhaustion will going to be, the higher the Tregs are. And that here you can see in the graph E, we look back, sorry. The higher the activation markers, higher the CD the Tregs altogether. So like basically Tregs are also a good indicator of telling that when the Tregs are higher, then your patients are going to have a higher immune activation, as well as is going to have a microbial translocation is going to play an important role, and exhaustion is also playing an important role. So all these parameters are essentially giving an indication that the process which we happen to really treat in this patient actually very complicated. It's not really, we are addressing the one issue, which is HIV viral load or CD4 cells, but that's not the whole story. His whole story is a little bit more complicated. Here we look back in the Tregs in the context of the markers of TIM3, which is the microbial translocation, and IL-17 and the peripheral TS17 cells against the Tregs, and again positive correlationship there. Now, in this particular slide, I'm showing you the relations of CD4 absolute counts, which is basically most of the patients are treated on the basis of what is the patient's CD4 T cells is, because that also defines them whether they are moving towards from HIV infection to AIDS clinical condition, as well as HIV viral load. So these are two standard markers which are used for the patient care. But as you can see from here, we started, okay, let's look back if we can, sorry. That the activation markers with the T cell counts. So the T cell counts go down, and then you have the immune activation also going down at the same time. So that means what it's telling us that the T cell counts are still a good marker, and it also gives the indication if there's not enough T cells, then you are not having enough activation there in the body. So they play an important role. We look back also these markers on the activation markers. You can look back on the both compartments, which is CD4 cells as well as CD8 cells. So we look back on both of these cells, and we have the same structure there. And not only that, but when we look back in the context of TS17 cells, the same story goes. The lower the number and the lower the T cell counts, the lower the peripheral TS17 cells in these patients. So this part of the study, I can conclude that our study supports previous observations that CD4 T cells immune activation and immune exhaustion persist among HIV patients despite stable and continued viral suppression. Our results suggest an important association of peripheral TS17 cells with persistent immune activation and CD4 T cell reconstitution in combination antiretroviral therapy treated HIV-infected patients. We have seen a positive correlation of Tregs and peripheral TS17 cells with each other and also independently with immune activation, indicating a synergy between immune activation, inflammation, and immune suppression, which also impacts outcome of CD4 T cell reconstitution in patients on combination antiretroviral therapy. Our study also reports that the combined analysis of multiple inhibitory markers appear to be a better predictor of immune exhaustion in CD4 T cells. And that's very important for us to know that when we look back these patients, even the cost is there, but we may be able to do better service for these patients. These findings from our study suggest that a better understanding of the nature and function of the cellular subsets associated with immune activation and immune regulation in HIV infection is needed. Additionally, novel strategies are required to target immune exhaustion, immune activation, and inflammation in order to accomplish satisfactory immune reconstitution and thus desirable outcome in HIV patients. Because as I stated before my talk, that we are missing certain aspect of that, and that's one of the reasons why we are not able to have the total cure of the disease, because these are the factors which we are missing it out. So we may have to come up with the treatment regimens which can also target immune exhaustion, immune activation, and inflammation. That will be able to complete the story about virus suppression in these patients and keeping them under the check. Future scope of the work, which is also work in progress, in order to accomplish satisfactory immune reconstitution and thus a desirable outcome in HIV patients, novel strategies are required to better understand and target inflammation, immune activation, and immune exhaustion. Now, this is one aspect of my story, and I think I still have 15 minutes. Okay, so I'm going to use this time to give you another aspect of my study in this area, because when we are studying it, we are also trying to see how best we can understand the pathogenesis and how best we can address this. The one aspect which we already talked about it, then also we look back that what are the things which can trigger the immune activation. And tobacco use has been also been considered to be one of the important factors, and there are a number of patients which are using tobacco, and tobacco is considered to be also an activator as well as immune suppressor. So we use this as a model to understand this mechanism a little bit better, and I'm going to share you some of the data from that one, and since we happen to have some time, and I hope that is of your interest. And then again, focus is again immune activation and immune function in the context of the parameters which I discussed to you earlier, the activation, exhaustion, microwave translocation. So what we know so far, the prevalence of tobacco smoking among HIV infected is as high as 70%. Cigarette smoke affects a wide range of immune function, thus impacting innate and adaptive host immunity. So knowingly, unknowingly, we are impacting on this thing. And if the patient happen to have HIV, then you are having like something additional to that, whatever is going on in the body. So clinical and experimental studies show that the cigarette smoke is both pro-inflammatory and immunosuppressive in these patients in general. Increased level of pro-inflammatory cytokines have been reported in chronic smokers as well as HIV-infected patients. T cells from smokers exhibit differences in proliferative responses to T cell mitogens, and I will show you some of the data on that area. Also in numbers and indicating defective T cell responses. Tobacco use has been known to significantly increase the risk of pulmonary diseases in HIV-infected subjects, along with many deleterious effects on antiretroviral treatment. So, as I said in my previous segment of my talk, that the activation and exhaustion and inflammation and microbial translocation plays out role, and I'm going to talk to you about that how tobacco plays a role in this process if we are combining with HIV infection. So here is in this particular slide, as you can see that here we have an HIV infection, and along with the tobacco use, you can see that it can lead to immune activation, and these are the markers for immune activation. I'm going to show you some of the data on that one. Immune exhaustion is also with the markers which are there. We are going to study that part, and inflammation, we are also going to look back in inflammatory cytokines in these patients, and we are going to say to show you that this also plays an important role, and we can study that part. And microbial translocation, which is defined by LPS and soluble CD14 markers, is also another good indicator to understand what's going on. And then on top of that, I'm going to demonstrate to you that when cells are exhausted, then actually they are not doing their work. And I'm going to show you some data there. So hypothesis is, despite virological suppression, the combination of smoking and HIV infection lead to chronic immune activation, thereby placing HIV-infected smokers at a higher risk of infection compared to HIV-infected non-smokers and HIV-uninfected age-matched controls. So this is the characteristics of the study participants, which you see. We happen to have HIV positive smokers. We have 25 of those patients. These are, again, we are talking about patient-specific data, not the animal data there. And HIV positive non-smokers, we have 25 of those ones. And then we have HIV negative smokers, which is control group, and HIV negative non-smokers, which we also have. They're 15 each. And we look back all these parameters in the context, like which are standard parameters. We look at the, the gender, age, the lymphocytes and other markers and viral load, and you can see that the patients happen to have less than 50 detectable limits of the virus in the HIV patients. So it's the same, similar to patient population which I discussed you earlier. They are under control. So clinically they are in considered to be like the HIV free, essentially, because it's undetectable virus. So in this particular bar graph, I'm trying to show you that the frequency of the cells. So here we have higher cellular markers of T cell activation in HIV infected and uninfected smokers. And you can see in this particular slide that defined by the activation marker like CD4, HLA-DR cells, and we are looking back the markers. This is smokers group and this is non-smokers, right? And that's HIV infected. And when we compare back with controls, these are the ones which are smokers negative HIV and smokers like HIV negative non-smokers. And we look back here with the just HLA-DR, and in this one we look back CD38, HLA-DR. Both of these are markers of immune activation. So we look back, and both are saying the same thing, that higher the smoking, the higher these patients are, the smokers are there in the HIV as well as in non-HIV patients, the higher the immune activation is. And here we have the CD8, because which is we look back both these cell markers, CD4 and CD8, in these patients. So I'm going to show you this data from both of these patient populations, both these compartments of the patient population, and address you that the higher the immune activation in these smokers. So smokers play an important role in terms of higher the immune activation. So then we look back also in the context of association number of cigarettes smoked per day, BMI, and alcohol drinks per week, which is not, I'm not going to cover that particular part of alcohol drink, but this gives you some scenario that what we have found. So I instead of spending time there, I'm going to say association of number of cigarettes per day, analysis of covariates of BMI and alcohol consumption per week. In all 4 study groups showed a negative association between BMI and CD4 immune activation, but not with alcohol drinks per week. The CD4 immune exhaustion was also negatively associated with the drinks per week. Number of cigarettes per day did not show any association with markers of CD4 immune activations. We did not observe any association between number of cigarettes smoked per day, BMI, alcohol drinks per week, and plasma levels of soluble CD14 and LPS. In addition to that, as I said in my earlier talk before starting this segment, that we also look back inflammatory cytokines on these patients. So we have the 4 groups which we have shown you earlier: HIV positive smokers, HIV positive non-smokers, and the control group, which is HIV negative smokers and HIV negative non-smokers. And we look back IL-1 beta, IL-6, IL-8, IL-17, anti-NR alpha, and T cells produce various cytokines upon activation. Therefore, we evaluated these plasma levels of pro-inflammatory cytokines in the study participants. Although differences between the smoking and non-smoking participants were not significantly significant, increased levels of circulating of these inflammatory cytokines were observed in the smokers of both groups compared to the corresponding non-smoking groups. So we also look back the microbial products in the bloodstream of smokers and compare them with the non-smokers. And here you have the markers of microbial translocation, which is soluble CD14. And we look back the frequency of the cells which are expressing these markers, the soluble CD14 in this cell population. And we look back in the context of the immune activation, which is CD4 positive HLA-DR CD38 cells, and we compare back with what is the level of soluble CD14 in these ones. We measured it, the positive correlationship, and we look back on CD4 and CD8, both compartment, B and C shows up there. Here we look back another microbial product, which is in the bloodstream of smokers with the immune activation. We have LPS, which is another marker used for the microbial translocation, and we compare back with the immune activation marker CD4 and CD8, and this has a positive correlationship there too. Higher levels of immune exhaustion markers in smokers compared to non-smokers. So here we have markers of immune exhaustion which are defined by the PD1, CD4 positive PD1 cells, and CD4 positive TIM3 cells. These are markers of immune exhaustion, and then also we have CD4 positive CTLA4 is another marker of the immune exhaustion, and essentially shows that direct relationship between those two. Higher the higher in the case of smokers, we happen to have a higher exhaustion, which can be defined by the compare back between the smokers and non-smokers, and we find that they happen to be higher immune exhaustion in these patients compared to the non-smokers. And this is the first one was the CD4, and now I have show you in CD8 compartment the same thing, which is PD1 and TIM3 and CTLA4. The markers of exhaustion in higher in the case of smokers versus non-smokers. And this particular slide shows you in the pie chart higher level of immune exhaustion markers in smokers compared to non-smokers. And I look back on the both compartments, CD4 and CD8, and you can see from here these are 4 groups of patients, and they are defined by these are HIV positive smokers. Like as you can see from this is the open graph, higher the numbers are here, and then again the one which is HIV negative smokers, which is this graph, and you can see that they are higher in all the cases altogether. So decreased frequency of now the idea was to see that we have identified so far that immune activation, exhaustion, and microbial translocation all playing an important role in this process. So when we say that the cells are exhausted, how we can define them? They are exhausted. They are not doing their job. So good idea there is to understand their functionality, what they are supposed to do it, whether they are doing it or not. So what we did that we have taken out, we look at intracellular cytokines essentially. So we isolated the cells from these patients. We stimulated them using the common stimulators, which is basically the markers which are used in the standard settings like CEF and SEB, which are used in. And we look back that once you stimulate these cells, whether they can produce IL-2, interferon gamma, and a granulation marker like CD107A. And if they do the job, that means they are normal cells, more or less. They can doing functionally a good job, what the God has meant or the system has meant to be, made it to be doing there. So we looked at that phenomenon in order to established our hypothesis that cells are exhausted, what we have shown so far with the markers and other things, and these cells are not only exhausted, they are also not doing their job, what they are meant to be doing it. So we look back that with the intracellular cytokines, and you can see from this slide that here you have where we took the PBMC, we stimulated with them CEF, and after stimulation, we look back intracellular cytokines, and we look back CD4 positive IL-2 production on these cells, and this is, you can see from here that the non-smokers happen to have a higher the response on these cell types. And it's not only in that particular compartment, but you look back interferon gamma, which is another good marker of these cells' productivity or their functionality altogether, that you can see that they happen to have a higher responses there. And we look back on the granulation marker, which is secreted by these cells once they activated. And then we look back not only CF, but we look back SEB, another stimulator. And we look back, it's not only that in particular this case, but in this case also we see the same phenomenon. That these are the non-smokers happen to have a higher cell responses and functionality than the other ones. Lower frequency of IL-2, and here we have the previous slide I showed you was on CD4 cells, and here we are looking at the CD8 cells. The same thing is we are seeing here, that what I was telling you earlier, that higher the non-smokers happen to have a higher response on the cell types versus the versus the smokers, and this all the parameters which we have studied there. So we can conclude HIV-infected combination retrovirally treated smokers who are virologically suppressed have higher levels of immune activation and exhaustion than HIV-infected age-matched non-smokers and HIV-uninfected controls. It still provides evidence that smoking is associated with compromised immune cell functionality. And the future studies, there are many questions which need to be answered, and the story is not complete so far, including whether immune activation could lead to opportunistic infections and accelerated application of the virus or increased damage to immune system of the smokers. And we are talking about the smokers as one example here, but in general we are talking about a patient as a whole, as a body. Future studies should be focused to understand the mechanism of immune activation and immune exhaustion and its impact on disease progression and its correlation with clinical outcomes in HIV-infected smokers. As recently suggested by our group, viral persistence is facilitated by immune activation. Therefore, strategies to contain immune activation, including control of microbial translocation, need to be further explored in these patients. With that, I would like to acknowledge the team of my members. And I have some people from Kerala also in my team. And these are my collaborators and some fundings from agencies which have been funded our work. And this is some of my team members. And thank you very much for your time.
Thank you, sir. Thank you for sharing your expertise and insights with us this morning. Sir, you have beautifully explained the delicate balance between the immune activation and T-cell exhaustion in HIV infection and its implication. We shall move on to the next segment of this program. I request you to kindly stay back on the stage, and I request the other dignitaries to kindly join him on the stage. I also request Mrs. Devi, wife of Dr. D.M. Vasudevan, to join the dignitaries on the dais. Father Terry Mulakara. I request Reverend Father Terin Mullakara, Assistant Director of Medical Education and Services, Jubilee, to hand over the memento to our esteemed speaker as token of our love and appreciation. I also request Shibu sir, our medical superintendent, to present a gift to our orator as token of our gratitude. Now we have presentation of a book entitled A Gentleman's Voyage, written by Dr. Kesia Kuruvela. I request Vasudevan sir to sign an autograph on the book and present it to our orator. As I said before, this book was written by Dr. Kesia Kuruvela. Dr. Kesia pursued her PhD under the guidance of Dr. D.M. Vasudevan. As a heartfelt tribute to her mentor, a true Guru Vandanam, she has authored this beautiful book entitled A Gentleman's Voyage. Thank you, ma'am, for this book. Let us capture this special moment together. May I request all the dignitaries on the stage to kindly join for a photo session. Thank you all once again. Before the vote of thanks, may I request the audience also to come to the center so that we can have a group photo together. I request the audience to kindly come to the center of the seats so that we can include all of you. എല്ലാവരും ഒന്ന് സെന്ററിൽ വന്ന് നിൽക്കണേ, ഒന്ന് നിൽക്കണം. എല്ലാവരും ഒന്ന് സെൻറ്ററിൽ ഒന്ന് വന്ന് നിന്നാൽ എല്ലാവരും നമുക്ക് ഇൻക്ലൂഡ് ചെയ്യാം ഫോട്ടോയിൽ. Thank you all once again. Before we move, I request Dr. Matthew John, scientist of JCMR, to propose the vote of thanks.
So today is a very special day for all of us. Professor D.M. Vasudevan Endowment Oration, the 8th discourse, happened here. And my duty is to say the final word of vote of thanks to all of you. And first, let me give a big thanks to today's speaker, Dr. Deshratn Asthana. Sir has delivered a very elaborate and scientific insight into the role of immune compromise in HIV infected patients and how the tobacco smoking activates that process. Sir, it's a privilege to hear you, and once again, thanking you for inviting our invitation and joining for this talk. Thank you, sir. And today's program is happened because of our great leader, our research director, Dr. D.M. Vasudevan. Sir and Devi Madam, both of them are here to grace this occasion. And thank you, sir, for making this a wonderful success again. Thank you, sir. And next vote of thanks is for our director of Jubilee Mission Hospital Trust, Reni Achan. Achan has blessed this occasion and made a very graceful speech and introducing the audience as well. Achan is very supportive to research, all our research initiatives. Thank you, Achan, for joining the session. And next vote of thanks goes to our Assistant Director for Medical Education, Father Terin Mullakara. Achan is busy around with a lot of important things happening in this institute in this golden hour. In spite of that, Achan came here and makes his presence. Thank you, Terin Achan, for your presence. And next thanks goes to our principal, Dr. M.A. Andrews. Sir, in spite of his busy schedule, has been attending this session wholeheartedly and supporting our research initiatives and activities as well. Thank you, sir. And next thanks goes to our medical superintendent, Dr. Shibu Kallivalapil. In spite of the busy schedule, sir is here, very much here, and actively supporting us. Thank you, sir. And next thanks goes to our pillar of this program and master of this entire research schedule here. And sir is the pillar. Dr. Piyavar is our research coordinator. And sir, your presence is very much for us. And thank you, sir. And Dr. K.G. Raghu, sir, happily accepted our request for introducing the speaker, and sir, thank you, sir, for making your presence. Thank you. And I would like to thank all the guests and the students of Professor Vasudevan, sir, for accepting our invitation and joining here. And next to all the students who come from various institutes of Jubilee as well as outside for making this program a grand success. Thank you, one and all. And Jubilee administration team, a very special thanks to all. I cannot make any individual names here. And thank you, the entire administrative team, for supporting our program a grand success. And thanks to the hospitality department, maintenance department, and the entire Jubilee team, and not the least, our thanks to the entire professors of Jubilee Medical College and Hospital who come here and make this occasion a success, and all the JCMR staff, students, PhD scholars, day in and out worked for making this program happening to this level. Thank you. One and again, all, and with God's grace, it all happened, and thanks Almighty for making this 8th discourse a grand success. Thank you all.
Thank you, sir. With this, we conclude this year's oration. Thank you all for joining us. Take care until we meet next year on 6th March 2027 for the 9th discourse of Dr. D.M. Vasudevan Endowment Oration. Please join us for the tea, which is served outside to your left side of the hall. Thank you so much once again.